Now we're going to turn our attention to translational and post-translational regulation. Now, we will note again that the majority of our regulation occurs at transcription. That is most energetically favorable. However, it can occur later. So, to occur at the translational level, there are these translational repressors. So, what is their job? To stop translation. Now, they can do this in a couple ways, but the ultimate goal is to stop the ribosome from interacting with the mRNA molecule. So you should remember in translation that we have this small ribosomal subunit that has these initiation factors bound to it. And they also have this 16S RNA sequence. So that 16S RNA is complementary to the Shine-Dalgarno sequence. And by binding together, they put that start codon, position it right where the P site will be. So there are molecules that will bind to this mRNA in this vicinity that doesn't allow the Shine-Dalgarno to interact with the 16S RNA of the ribosome. Therefore, they don't interact, they can't bind, we can't have translation. There are some molecules that bind to the mRNA in a region that's not in the Shine-Dalgarno, and they cause our mRNA to take on a structure that stops the ribosome from being able to bind. Okay. Another way is through antisense RNA, and that should sound familiar to you because we talked about this with the non-coding RNA molecules. So these antisense RNAs are complementary to an mRNA molecule, and again, they physically block it from binding or interacting with the ribosome. This particular one functions in osmoregulation in E. coli. So OMF is a gene, okay, it's an mRNA and it's a protein. The name applies to all three, but they are different molecules. And this OMF is made when we have low osmolarity. Well, when our osmolarity increases, we make MIC-F, and MIC can bind to OMPF to stop it from functioning. Okay, stop the translation by stopping the ribosomes. Now, post-translational regulation can occur in a couple ways. One is feedback inhibition. So a lot of the metabolic enzymes have this feedback inhibition. So there's a pathway where enzyme 1 makes the substrate for enzyme 2, 2 makes the substrate for 3, and so on. And so this goes on and on and on. We get our final product, and the idea is to ramp up production of it. Well, once we get so much, this final product can serve as an effector molecule to the first enzyme. And if it binds there and changes the shape, we can no longer go through this process. Or we can do post-translational modifications. So we can modify our proteins in a way that we stop them from working. So if you think back, we can do things like cleavage. We can put on ubiquitin, which targets it for degradation. We can methylate it, acetylate it, phosphorylate it, and these are all things that are reversible, but that allow us to turn on and off proteins as needed. Okay, so those are translational and post-translational modifications. So the last thing we're going to talk about is a riboswitch. So this riboswitch is interesting because it's an RNA molecule that can exist in two forms. So it can be active or it can inhibit gene expression. And this is regulated by a small molecule. And so it can go back and forth between the two depending on if this molecule is available or not. It's important to note, 3 to 5% of all bacterial genes are controlled by this riboswitches. And these are things like how they make purines, amino acids, vitamins, and other essential molecules. So they are dictated by riboswitches. Riboswitches exist in bacteria, in eukaryotes, archaea, algae, fungi, and plants. And now they can also control transcription, translation, mRNA stability, and splicing. Okay, so these riboswitches are very important. They spread out over different organisms and over different processes. So let's look at the riboswitch for transcription. So we have our two conformations, and we're switching back and forth depending on if we have a small molecule And in this case, our molecule is TPP, or thiamine pyrophosphate. Sorry. So when it's low, we have our active molecule. When we have high TPP, we're now going to inhibit. So how do we inhibit? So here we have an anti-terminator stem loop. Well, over here it's a terminator stem loop. A stem loop followed by a uracil-rich sequence. We know that this is code for a row-independent termination. Okay, so we switch back and forth. If we have high TPP, it's attenuated and it stops. It doesn't get to go through the whole process. This is dictated by a stem loop followed by a uracil-rich region. In translation, it's a similar process. We have our TPP, so it's either is the shine doggone node accessible or not. If it's not, we can't have translation. If it is, we can. We can go back and forth depending on if we have our